So both Michael and Bill have defined their terms in slides as they started and introduced their subjects. I've not done that. I think I'll try to do it verbally then. And I think the key issue is what is targeted. So at its very best, then we would know that there may be one mechanism that drives a cell to be cancerous or a disease to be cancerous in an individual, and you conceive a molecule that will inactivate that mechanism, and things will return to normalcy. A different step up from that is that with the tremendous advances that have been made in biology over the past 2 decades, we've certainly come to realize that there are commonalities within certain cancers. Say if you take CLL, not all CLL is driven by the same mechanisms, so it needs separate therapies, so that individuals who have characteristics that are one driver would get a therapy that wouldn't be effective in another group of patients with the same disease. So that's where we are today with targeted therapy, and you'll hear the term personalized medicine, and then it's personalized to the extent really of our knowledge. We don't want to address ourselves because we have a lot to learn about individuals. I was listening to a man on Saturday afternoon at a symposium here, and he's talking about sequencing the DNA of single cells, and using that within 10 years, that will be possible. And this is a person who's come through on these predictions before. So, but this is the state of the affairs today with targeted treatments. What we're hoping to do is to identify individuals whose cancer is driven by a particular one or set of drivers, and then try to attack those. And that collectively is known as the pathophysiology of the disease. Really, what is it may not be the thing that started the disease, but it may be rather it's the mechanism that keeps it going. So I'm going to speak about 5 different aspects of the pathophysiology of chronic lymphocytic leukemia, and approaches that are being taken now. Some of these are in phase 1 or 2 studies, and some we're just hoping to move into the clinic, and then and bringing these forward. So I like to start by asking a question, and then I know whether I'm doing well or not. If I can provide myself or my group can provide an answer to a particular question, and of course you hope that the question is important. Okay, so what aspects of pathophysiology of CLL are associated with failure to respond to current therapies? So current therapies, let's just take, for example, frontline therapy, which is now accepted as fludarabine, Cytoxan, and rituximab. This large multi-center randomized trial was published last week in Lancet. And we know that FCR, as it's known, gives a survival advantage over the previous therapy, fludarabine and Cytoxan, that was a major advance. Both of those, I would say, came from Dr. Keating's clinic. And then were subsequently demonstrated in a randomized trial in large organizations that have the capability for doing that. Well, one of the aspects of pathophysiology of CLL that's been known for some time is that CLL overexpresses proteins that allow it to circumvent cell death mechanisms. So these are called pro-survival proteins. And the CLL cells generate an excess of that, and those pro-survival, it sounds very simplistic, but this is how actually we talk about it. Pro-survival proteins neutralize the activity of proteins that are going to kill the cells by digesting the DNA and various components of the cell. So it's a plus and a minus interaction of cells. So what we've come to recognize is that some of these proteins have very short half-lives. They have to be continually made in the cell. So we're reasoning then, if we block the synthesis or the production of these pro-survival proteins, perhaps the concentration in the cell will diminish, and therefore the pro-death or the death proteins will dominate, and that will that will affect the death of CLL cells. And this mechanism of death is known as apoptosis. So it's used. It's just generally used in this context. So we have several potent inhibitors of the couple of synthesis steps in the making of proteins, because as you know, proteins are coded from DNA. Proteins are coded from DNA, and then that DNA is translated or transcribed into an intermediate, which is another nucleic acid called RNA, and then proteins are made from that. So if we start then with, this is the rationale, an inhibitor of transcription, this first step, then this should lead to a diminishing of the RNA molecules. So these are needed to make proteins, and there's a one-to-one correlation between the amount of RNA and the amount of protein that's present. So then, secondly, in this sequential pathway, then a translation inhibitor would block that step and then diminish the amount of these pro-survival proteins that are present. So the pro-survival proteins in the next indication are bound to these little orange crescents here, which indicate these are the pro-death proteins. But with fewer pro-survival proteins, then you see that there are more pro-death proteins that aren't neutralized. Their activity isn't neutralized. This is real. We can see this in CLL cells during therapy. And then, of course, subsequently, then that is seen to be associated with cell death. And we have strong proof of principle of this using primary CLL cells in the laboratory, and we've extended this to the investigation of a couple of these inhibitors in the clinic. Well, so then there are two places that we can stop, and we can think that maybe we could combine these. So that hasn't been, that's been done in the laboratory, and it is effective, and we're looking forward to that in the clinic. And we have elements, we have inhibitors here and here in the clinic, and it's a job to get them together. So an alternative approach to this overexpression of these pro-survival proteins might be to take this complex of the pro-survival and pro-death proteins and pry them apart to liberate the pro-death proteins. Well, then there are several molecules that have been made, because we know what the molecular structure of these proteins is, and there are people who are smart enough to make these proteins. So then if we start at just with infusion of these proteins that will interact between the pro-death, pro-survival proteins, and they do that job, and then what they do then is instead, the pro-survival proteins, instead of binding to the death proteins, this compound, this new drug, will do that and then liberate the pro-death proteins, and then you get the cell death that you're looking for. So these are strategies that are coming along at our clinics and elsewhere.